The experiment that we are going to perform on International Space Station regards the use of microchannels and microparticles to study the transport phenomena in a confined environment. My research team has been focused on implantable drug delivery system for the controlled administration of therapeutics for a extended period of time for the treatment of chronic pathologies. When we scale up from the nanoscale to the microscale, obviously the mass becomes relevant. So while molecules at the nanoscale are not very much affected by a gravitational force, gravitational field, microparticles start having a relevant mass. Therefore, we need to remove the gravitational force from the picture in order to be able to capture what really happens into the diffusive transport. This is the reason why we intend to study this in microgravity onto the International Space Station. So I clearly envision how this system and this experiment that we are performing in International Space Station will give us the right insight to develop the new generation of our implantable system, knowing how molecules actually diffuse through the nanochannel. We really believe that this kind of insight will be instrumental in moving forward and developing the new generation of devices. By having a theoretical understanding of what really happens into the nanochannel, we will be able to bypass a lot of these work and efforts and actually focus directly on what is going to be much closer to the final product. This IP, intellectual property, has been licensed by a company called Nanomedical System, which is based in Austin, Texas. And Nanomedical System is now moving forward toward clinical translation with, again, an implantable device for controlled administration of therapeutics over an extended period of time. So we are really thinking that our research will actually have some benefit for the population, for patients in the near future. We expect to observe a diffusive transport of negatively charged particle to be hindered by the constraining microchannel, while instead for positive charged particle we expect to see a much faster diffusion with accumulation of particle along the channel surface. We will receive images captured on International Space Station in real time. This will allow us really to understand immediately what is happening to our devices, our microchannel devices, and to tailor the experimental analysis based on what we are observing. We have been funded by CASES for a 5-year project in which we will test an implantable device that can be remotely controlled through Bluetooth RF frequency. CASES was particularly interested in this technology because it can be used as an enabling system, enabling technology for study with mice and animals on International Space Station for an extended period of time. On International Space Station, one of the biggest limitations is the fact that you cannot handle the animal freely as well as you do here down on Earth. Therefore, you cannot administer drug at different time points, and you are really limited in the spectrum of research and experimentation that you can do. I believe there is real potential into this investigation that we are performing, as well as the future investigation that we are scheduled to perform for the implantable drug delivery system.